So I'm Romina Henriques. I work at the Division of Genetics of the Biochemistry, Genetics and Microbiology department here at UP, and I am a molecular ecologist. So I try to understand how oceanographic features influence evolution of marine fishes, and I am particularly interested in understanding the interplay between evolution and fisheries and how that affects the evolutionary resilience of our marine species. And now with climate change, South Africa is a hotspot for climate change. In terms of marine environment, things are changing very quickly, much faster than it was predicted. And what's going to happen is, it looks like we're going to have a contraction of our temperate environment, which is the South Coast. So species that are reliant on the South Coast are not going to fare very well.
So my name is Mannda Ndou, and I am doing a PhD degree under the supervision of Dr. Romina Henriques under the Department of PGM, and my PhD project or research is about the conservation genomics of an endemic fish species that is called the red romaine that is found in the South African coastline. Red romaine is a significant contributor to the South African economy through the fisheries, and another thing is that it's a beautiful fish species and is endemic to South Africa, which makes it to form part of a valuable part of our heritage in South Africa.
Mannda's research is part of a stream of research that we are starting now. It's called climate change genomics, which is genomics to understand the consequence of climate change. So species have three options with climate change. You move, you adapt, or you die. I'm not sure dying is an option, but it is an outcome. But not movement is equal, because you might get individuals moving into a new environment and they are completely unsuitable for the new environment. Therefore, they move, they die, they don't establish. We are trying to use genomics to see if we can predict which individuals will be able to move and adapt and stay and proliferate. The genome is super important because we want to know what genes might be involved in these mechanisms. And for marine species, as I say, compared to land research, we are 10 years behind. Being optimistic that we are 10 years behind. We don't have the genomes, we hardly have the species sampled. So this is, right now, South Africa is one of the few places that is doing it.
Basically, having a reference genome is like having a blueprint where one can map things upon. And having a red romaine genome will be like having a red romaine specific blueprint where all the sequenced sample data will be mapped to configure all the variations and some important genes that are only found within the red romaine that we could not configure out if we were using a genome from a different species. This is going to be an essential genome that will enable us to understand fully the biology of the red romaine.
Actually, we have very few genomes for marine species in South Africa. It's not a South African bias, it's a thing, because there's very little investment in marine species. And we need to build these genomes. And there are several ways to do it. The long reads give us just that extra advantage because it allows us to sequence much longer parts of the chromosomes, which means when the time comes to assemble these reads into prototype chromosomes, it will be easier. We will have more information. And it will give us a better idea of contiguity already, what genes go next to each other. So it's a new technology, and it also gives my students new skills on a new field, which is very exciting, but also it gives us more info.
So my project aims on assessing or investigating the importance of marine protected areas in conserving the evolutionary resilience of the red romaine species across and even between marine protected areas and the overfished areas. But it goes to an extent that we also want to assess the genetic diversity across all the populations that are found across the South African coastline, and also the genetic connectivities of those populations.
Marine protected areas are designed generally, especially in the past, you wanted to protect something. It can be a region, it can be that you want to protect the spawning ground of something. And we know that these marine protected areas need to be well connected because fish swim. It's a characteristic of fishes. But not all fish swim very far, and you need to have them relatively close to each other, right? So you keep the connectivity, and then they do a thing that we know they do well, which is the spillover. You create an area where fishing is reduced or not allowed at all. Fish are allowed to grow bigger. Bigger fish produce more eggs, and then you start having spill over to the sides and you replenish the areas outside of the marine protected areas. What we don't really know, or we have fewer evidence supporting, is what will they do in terms of protecting genetic diversity. Genetic diversity is fundamental for a species to evolve. Think about it as the building blocks of a species. If a disease comes, you'll have some individuals that might be able to survive it, for example. But now we are in a context where everything is changing. The world is changing. It's not coming, it's here. And these animals had millennia to adapt to their conditions, and our conditions are changing very quickly. So we do need a lot of genetic diversity to keep up. When you have commercial fisheries, you are taking part of the population. So you are already removing part of the genetic diversity contained in this population. Now let's assume that we will never stop fisheries, which is a very healthy thing to assume because we need protein. So we have to find a balance. But you can only find that balance if you know what's there. And if you don't know what's there, there'll be no balance to be found. We can do everything we want, but we're going blind. My research is not going to prove marine protected areas are useful, but it's a piece of evidence that is fundamental to regulate fishing and to regulate the marine protected areas.